A very warm greeting to all of you. It is my great pleasure to share with you the latest update on our project, which is funded by the GlobalSeaweedSTAR on exploring genetic diversity of Kappa ficus and Eucheuma in Indonesia for crop improvement. The project member consists of Professor Guy Gachon, Professor Juliet Brody, Dr. Ziwan Po, Dr. Eka Sunavidi Prasadevia, and Dr. Janina Brecker. Okay. What are the rationale behind that we carry out this study? This is mainly that based on the existing 3 report on genetic diversity of eukaryotic from Indonesia, but this has rather limited as regard about the depth of the sampling. Indonesia is very important because actually Indonesia is the largest world production for carrageenan phyte. And based on the 3 reports, it shows limited genetic diversity between the cultivar, and this will affect the overall productivity over time. Given that genetic diversity is very important in creating resilience both in the ecosystem and living organism. Without large pool of genetic diversity, the industry is vulnerable to disease outbreak as well as climate change, and subject to frequent outbreak of devastating diseases, in particular ice and epiphyte infestation. Therefore, there's urgent need to look for greater diversity and for more haplotype of Kappa ficus and Eucheuma to be used for cultivation. The main objective of this study were to achieve an unprecedented depth of sampling of cultivated and wild Kappa ficus and Eucheuma population in Indonesia, with emphasis on their diversity hotspot, the Coral Triangle. To establish the molecular taxonomy based on genetic marker includes the mitochondria encoded gene or spacer, for example COX1, COX33 spacer, as well as chloroplast encoded marker, which is RBCL gene. This study will fill in the gap in the current ongoing project of GlobalSeaweedSTAR, where the same methodology is deployed in the Philippines, Malaysia, and Tanzania. Now I would like to share with you the progress and key result of this study. This project has achieved an unpredicted coverage as regard about eukaryotic sampling from farm and also natural population across the Indonesian archipelago from Sumatra in the west to Papua in the east, and this include Java, West and East Nusa Tenggara, Sulawesi, Kalimantan, and Papua. The fieldwork was carried out at 47 sites with a total of 922 samples were collected from 46 sites where no cultivation was carried out during visit to Moluccu. Out of the 40 site sample, 11 are natural population. Okay. Based on the study, we found that only 3 species were used as cultivar in Indonesia, which is Kappa ficus aureata, Kappa ficus striatus, and Eucheuma dendriculatum. In contrast, natural population sample consists of only Eucheuma dendriculatum and a putative new species of Kappa ficus. However, given that the sampling of wild specimen was limited by time and lack of diver, the diversity of wild Eucheuma in Indonesia is likely underestimated. Because based on the study carried by Weber and Bose in 1913, they are the Eucheuma cottoni, or currently known as Kappa ficus cottoni, was first described in 1913, and in 1928, Weber and Bose described several new species of Eucheuma, which consists of Eucheuma leuvini, Eucheuma crustiforme, Eucheuma adherens, Eucheuma diatotonum, Eucheuma cervico, Eucheuma compressum, and Eucheuma simplex, and there, but we did not encounter that in the current sampling. Based on the result, All Kappa ficus aurelia and striatus collected belong to a single strain or haplotype, as we can see from the table, which is based on COX-2 spacer as well as COX-1. This indicates a severe lack of genetic diversity between each species. As for Eucheuma denticulatum, there are slightly more in terms of number of haplotypes were identified, and 3 of the COX-1 haplotype are novel haplotype. At the same time, based on the molecular analysis, it shows that there's a putative new species from the wall, and currently the work is ongoing in examining the morphological features of the collected sample. Some of the potential impact for the seaweed industry derived from this study are, based on the output of this project, it shows that there's very low genetic diversity among cultivar despite the widespread geography distribution. We intend to provide recommendation through a peer-reviewed publication for the local industry to consider the need of diversify the cultivar, in particular introduction of new genetic resources from local indigenous wall stock, which require more in-depth exploration. This is possible because based on study, there's a putative new species as well as some new haplotypes have been discovered through this study. This is vital as a narrow gene pool generally represents little resilience to threat of emergent disease, pests, and climate change impact. Besides that, seaweed industry also help to address SDG number 1, which is non-poverty. As we can see in the news that seaweed farm help Balinese survive pandemic, so it is a very important source of income for the coastal community. The outcome of this study will benefit first and foremost the farmer, the majority of whom are very poor and do not have much knowledge on the best strain for yield as cultivar. Introducing more genetically diverse broodstock with high productivity is crucial to reduce the risk of extinction and safeguard livelihood. The seaweed industry not only addressing SDG 1, but it also addresses other SDG as well. For example, this project has enabled a better and current knowledge of the genetic diversity of eucheumatoid in Indonesia, which address SDG 14 on life below water, and identification of potential new cultivar that are resilient to disease and climate change are essential for the sustainability of the industry. Besides that, close collaboration with the local partner, in this case Mataram University, provided much needed information on cultivar and natural population in Indonesia, which is the largest carrageenophyte producing country, to the GlobalSeaweedSTAR Consortium for a global analysis and in part address SDG 17 partnership for goals. Capacity building is also one of the emphasis of the project. During the course of the project, researchers in UM provided training to 2 undergraduate students from University of Mataram in molecular analysis of eucheumatoid, including DNA extraction, polymerase chain reaction, or in short PCR, gel electrophoresis, and herbarium preparation. 2 temporarily technical assistants which were recruited for the project were also trained in the same aspect. To conclude my talk, I would like to thank a few people for their kind assistance while we carry out this project, which is Dr. Tan, Mimi, as well as Mariana. With that, I would like to thank you for your attention.